Okay, in this unit, we're going to be looking at the chromosome formation, how these chromosomes form, specifically their parts, um, what we can learn from them, those types of things when we're looking at how they deal with cell division. So let's go ahead and get started with this. We've talked about before how the cell cycle and cell division occurs differently in different types of organisms. Okay, we know that those two broad categories, prokaryotes and eukaryotes, we know that eukaryotes, they have those organelles, so they use that cell cycle that we looked at in the last unit. They go through mitosis to divide the nucleus. They have the, they go through those stages like interphase, cytokinesis, those types of things. For our prokaryotes, like our bacteria, are more simplified. But no matter what type you're looking at, the end result is of these new cells, there must be a complete copy of genetic material that's an exact copy that's going into these new, going into the new cells that are being made. Okay, we've looked at the eukaryotic cell division. Here are some. Here's a way that prokaryotes, like like bacteria, can go through cell division. And I've mentioned this word before, but the process is called binary fission. It's a form of asexual reproduction. Okay, and what happens is you have this single-celled bacteria. It's able to divide, split itself in half. Copying its DNA or copying its genetic material, ribosomes, those types of things. And when it splits itself in half, it produces two identical cells as a result. Okay, so one bacteria can split itself in half, producing two identical bacteria as a result. This is binary fission. Two quick stages: DNA is copied, other materials are added to make sure that there's complete sets on both sides, and then kind of like in the animal cells, that cell membrane is going to pinch together and split that one bacteria cell in half into two. Here's a good little picture representation of how it works. We'll get into this in more detail when we get to bacteria, but here's your representation. You have your one bacterium. Okay, it's going to copy its DNA or its genetic material. Once it does that, the bacterium cell is going to push both copies to the sides of the cell, and then it's going to split in half, forming two identical bacteria as an end result. That's just straight binary fission. Not a whole lot going on. We know when we look at eukaryotic cells, during that interphase process, those chromosomes are going to form to package all of that DNA into these smaller packages. Okay, so these chromosomes are going to form as a result of that DNA coiling up, wrapping around proteins. They're going to form two structures that make the chromosomes called chromatids. We call them sister chromatids, and they're exact copies. Each chromatid is an exact copy of itself. They're attached in the middle, okay, with the structure called a centromere. And then these chromatids become separated during cell division. Okay, that's how this genetic. When we've looked at that through metaphase and anaphase and all those processes. Here is a computer illustration from an electron microscope of the structure of a chromosome. Once again, you see here is one sister chromatid, here's the other sister chromatid, and then here is the centromere. And then how it works, you take your DNA, okay, you have these proteins that the DNA is going to coil around, and it's going to bunch it up into really, really small packages. Okay, once it does that, then those coils get wrapped around even tighter, kind of like a spring, bunched together kind of like a spring, and then they form these chromatids that go into that form the chromosome. Here's another shot of a chromosome with an electron microscope, and you can see all the coiled up packages that form each chromatid. Okay, so let's start getting into our chromosomes. How many we have, that type of thing. We know what somatic cells are. We've talked about them. They're any type of cell that makes up our body that are not sperm and egg, which are those gametes, those reproductive cells. So all of our somatic cells have 46 chromosomes, 23 pairs, if you will, 2 copies of each. So they've got 46 total chromosomes. Each of those pairs, okay, and here's a nice picture of our chromosomes, and we'll get to what this is called a karyotype. We'll get to that later. But each one of these pairs, like here's pair number 1, pair number 2, and so forth. Each pair is made up of something called homologous chromosomes, which means that they're similar in shape, they're similar in size, they're even similar in the genetic material that they contain, like what they code for. One comes from mom, and one comes from dad. So they're homologous because they code for the same thing, they're very similar in shape and size, but just one comes from one parent and one comes from the other parent. Okay, and that makes up all 46 of our chromosomes, one from each parent, and they're broken down into groups of 2 based on homology, or how they're similar. Okay, so if you look at this picture, here is one set, there is one set, all the way down to the very end, and you got one from mom and one from dad in each one of those sets, in each one of those pairings. All of our somatic cells are diploid, which means they have 2 sets of chromosomes in them. Okay? Diploid means it has 2 sets of chromosomes inside the cell, inside the nucleus of the cell. So all of our cells are diploid, which means we have 46 total chromosomes, 1 set from each parent. Our gametes, our reproductive cells, the sperm and egg, are going to be considered haploid. And haploid means there's only 1 set of chromosomes within the nucleus of that cell. Okay, so within the nucleus of the sperm or the egg, there's only going to be 1 set of chromosomes, which means there's only going to be 23. Okay, so they're going to be considered haploid, which is important because if you think about reproduction, what makes a fertilized egg and eventually what makes an organism is the sperm and the egg coming together to form that fertilized egg. Well, if the sperm and egg each had 46 chromosomes, that means that the end result would be a new person being born that's going to have 92 chromosomes, and that's way too many. There's going to be a lot of complications. You don't want that many. You want 46 total. So that means that the sperm and egg are going to have 23. They're going to be haploid. Okay, fertilization occurs, right? You have your sperm and egg fused together. Okay, once fertilization occurs, once the fertilized egg has formed, it is going to turn into a diploid zygote. It's going to be called a zygote once the sperm and egg have formed or have merged into a new structure. The zygote is going to be diploid, which means it got 23 chromosomes from the sperm cell and it got 23 chromosomes from the egg cell, meaning that it has a double set, or 46 chromosomes within its cell now. If you're looking at all 23 pairs of chromosomes in a somatic cell, 22 of those pairs are going to be called autosomes, which means that they determine every type of characteristic except the gender. Okay, so once again, looking at these 23 pairs of chromosomes within one of our cells, with one of our somatic cells. 22 pairs of those chromosomes are called autosomes, which means autosomes are the chromosomes that make up every characteristic of us except our gender. Then there's going to be 1 pair of chromosomes that are going to be the sex chromosomes, which code for gender, whether you're male or female. In humans, we use X and Y. We label them X and Y chromosomes to categorize gender. Okay. If it's a male that you're looking at, the male carries an X and Y chromosome, meaning that they're going to be male as far as the species goes. And then the females are going to carry 2 X chromosomes, but no Y chromosome. So let's look at these karyotypes. A karyotype is a photo, an actual picture of what your chromosomes look like, and it gives you a snapshot of it gives you a snapshot of a lot of things, but it lets you determine any type of abnormalities that are associated with chromosomes. Okay. If we look at this picture, for example, you should have 2 sets along every one of these until you get down to the 23rd pair. Okay, so there's 2 here in chromosome 1. For chromosome pair 2, there's 2, and so forth. Notice when we get down to chromosome number 21, there should be 2, but there's actually 3 in this particular karyotype, this particular photo of karyotype, which means this person actually has an extra chromosome on the 21st pairing. Okay, if you have extra chromosomes or if you have missing chromosomes, that's where you get to these chromosomal abnormalities, and that can lead to genetic disorders. This particular genetic disorder that you're looking at, where there's an extra chromosome on the 21st pairing, that's called trisomy 21, and that's the cause of Down syndrome. This particular karyotype is showing a person that has Down syndrome, and they have 2 X chromosomes for the sex chromosomes, which means they're female. So this is a female that has Down syndrome. So you can notice a lot. You can determine a lot of genetic disorders by looking at a karyotype, if they have extra chromosomes, if they have missing chromosomes, and then obviously if it's male or female, you can notice that by looking at the last pairing here, if it's 2 X's or if it's 1 X and 1 Y.